Hi everyone. Welcome. Welcome to this quiet, soft space. Wherever you happen to be right now. Perhaps you are lying in your bed, settled beneath a warm blanket, with the room around you gently darkened. Perhaps you're in a comfortable chair, a pillow behind your head, legs stretched out, the day's responsibilities set aside for now. Or perhaps you're somewhere else entirely, somewhere private, and still, and yours, wherever you are. Whatever the hour, I am very glad you are here with me tonight. We're going to spend a long time together, a slow, gentle, unhurried stretch of time, and we're going to spend it exploring one of the most quietly wonderful subjects in all of science. We're going to talk about DNA, deoxyribonucleic acid, the long, patient, winding molecule that lives inside nearly every cell of your body and carries within it the instructions for almost everything that you are. And we're going to take it very, very slowly. There is no hurry here, not even a small one. There is nothing to accomplish tonight, nothing to remember, nothing to get right. We are simply going to let the words come, one after another, like waves arriving at a quiet shore. Unhurried, consistent, and soft. If the science is interesting to you, you are welcome to follow it as closely as you like. And if the words begin to blur and blend into something warm and sleepy, that is just as welcome. That is, in fact, perhaps the most welcome thing of all. If you drift away before we reach the end, please know that is not just acceptable. It is a beautiful thing. It means the words have done exactly what they set out to do. The science will still be here, patient as always, whenever you want to return. So, before we begin, let's just take a moment to settle. Let your body find its most comfortable position. Let your shoulders drop, if they've been holding themselves up, without your noticing. Let your jaw relax. It is very common to hold tension in the jaw without realizing it, and releasing it feels gentle and good. Let your hands rest somewhere comfortable without holding on to anything. Let your belly soften. Let your feet and legs go heavy. Take one slow breath in through your nose. Hold it just for a moment, and then let it go slowly through your mouth, and again in and out. The body already knows how to do this. The breath has been arriving and leaving since before you were born. It knows its rhythm. Let's let everything else follow that rhythm, the slow, patient, easy rhythm of the breath. And now, very gently, we can begin. Let's begin at the very beginning, at the simplest, most foundational thing, and we'll take each idea slowly, letting it settle fully, before we move to the next. DNA is a molecule. That is the most important and most basic thing to understand at the start. DNA is a molecule, a structure made of atoms, bonded together in a particular arrangement, and it exists inside the cells of living things. Every bacterium on the planet, every flowering plant, every fish in the ocean, every insect, every bird, every mammal, every human being, all of us, all of life as we know it, uses DNA to store genetic information and to pass that information from one generation to the next. And we can take a moment to notice how remarkable that is. One molecule, shared by all known life, the common thread, running through billions of years of biological history. Fact 1. DNA stands for deoxyribonucleic acid. It is found inside the nucleus of nearly every living cell on Earth. In the human body, almost every single cell, from the outermost cells of your skin, to the cells lining the interior of your stomach, to the delicate cells deep within your brain, carries a complete copy of your DNA, the full set of genetic instructions. That makes you who you are. That is a remarkable thing. If we let it sit gently, in the mind, every cell, a complete archive, every cell carrying the same full set of instructions. Instructions that fill, not one page or one book, but something more like a library, and that library is copied again and again, in trillion after trillion of cells. The same text, everywhere, all at once. For now, let's just hold the simplest version of it. DNA is a molecule. It lives in the cell. It carries instructions. Fact 2. If you were to take all of the DNA from a single human cell and carefully unwind it, gently straightening out every coil and fold, and then stretch it out into a single, perfectly straight line, it would be approximately 2 meters long. That is roughly 6 feet, taller than most adult human beings. And all of it is packed inside a cell, so small that you cannot see it. Without the aid of a microscope, 2 meters, all of it wound up, folded upon fold, inside something invisible. There is something very deeply calming about that image, if we allow it to settle, the idea that within something so small as to be invisible to the naked eye, something so vast and so orderly is folded up, patient and complete, waiting quietly to be read. Fact 3. The human body contains approximately 37 trillion cells, and nearly all of those cells carry that same complete set of DNA, the same 2 meters of instruction, copied in almost every single one. If you were to take all of that DNA and lay it end to end in a single unbroken line, it would stretch from the Earth to the Sun and back more than 60 times. More than 60 times. The distance from the Earth to the Sun is approximately 150 million kilometers. So we are talking about a total length of DNA in your body of something approaching 20 billion kilometers. The number is so large that it loses its shape and becomes something more like a feeling, the feeling of something vast, something almost beyond imagining, tucked quietly inside your body. As you rest, we don't need to hold that number tightly. We can simply let it rest there, like a large, smooth stone, placed quietly at the bottom of a still pool. It sinks gently and settles, and the water grows calm above it, and we can take a moment to notice, gently and without any particular urgency, how extraordinary it is that we are made of such things, that inside this body, right now, at rest, in the quiet, there is an almost unimaginable quantity of information, organized, folded, and carefully maintained, in structures smaller than anything our eyes can resolve. Now let's think, slowly and gently, about what DNA actually looks like, because DNA has a very particular, very elegant shape, a shape that scientists spent years working to understand, and when they finally saw it clearly, when the evidence came together, and the structure became undeniable, it was recognized, almost immediately, as something extraordinary, not dramatic, not complicated, just quietly, almost perfectly beautiful. In its simplicity, the shape of DNA is called a double helix. Let's take a moment to picture that, gently, without any effort. Imagine a staircase, not an ordinary, straight staircase, but a long, spiral staircase, the kind you might find winding upward inside an old lighthouse, or curling up through the center of a grand library, a staircase that curves in a slow, continuous spiral. As it rises, the outside rails of the staircase curve too, winding around and around, following the spiral, all the way up. Now imagine 2 of those staircases wound together, not side by side, but actually twisting around each other, their rails interleaved, the whole structure forming a long, continuous, graceful, double spiral. That is the double helix, 2 long strands, winding around each other, in a slow, elegant, continuous spiral, simple and beautiful, and stretching in your cells for a length that would measure nearly 2 meters, if uncoiled. Fact 4. The double helix structure of DNA was described in a scientific paper published in April 1953 by James Watson and Francis Crick at the Cavendish Laboratory in Cambridge, England. Their work built upon the X-ray crystallography images produced by Rosalind Franklin and Raymond Gosling at King's College London, which provided crucial evidence about the dimensions and arrangement of the molecule. Their paper was published in the journal Nature and was only about 1 page long. 1 page. It is worth pausing on that for a moment. One of the most significant scientific discoveries in the history of biology, a discovery that would go on to transform medicine, agriculture, forensics, and our understanding of life itself, described in a single, quiet page. Good science often finds a way to say important things simply. Fact 5. The 2 strands of the DNA double helix run in opposite directions to each other. Scientists use the word and a parallel to describe this. If you were to walk along one strand of the molecule from left to right, you would find the chemical structure oriented in one direction. Walking along the other strand from left to right, you would find the structure oriented in the opposite direction. It is a bit like 2 rivers flowing side by side, parallel, close together, connected, but each one flowing in the opposite direction from the other. Antiparallel, 2 flows, side by side, moving in opposite directions. It is a technical word for a structural feature, but the image it evokes is a simple and gentle one. Now let's think about what holds those 2 strands together, because the 2 strands don't simply float near each other. They are joined along their entire length by a series of small chemical connections, and those connections are what form the steps of that spiral staircase. Fact 6. The rungs of the DNA spiral staircase, the steps that connect the 2 strands and hold them together, are made of pairs of chemical molecules called bases. There are 4 types of bases in DNA: adenine, thymine, guanine, and cytosine. Scientists often refer to these simply by their first letters: O, T, G, and C, just 4 letters. The entire alphabet of the genetic code, the complete vocabulary of all hereditary information for all known life on Earth, written in just 4 letters: O, T, G, and C. There is something very restful about that kind of simplicity. Not 10 letters, not 100, just 4. And from those 4, the extraordinary variety of life, every form it has ever taken, across billions of years, is generated. Fact 7. The 4 bases do not pair randomly. In DNA, adenine always pairs with thymine, and guanine always pairs with cytosine. This is called complementary base pairing, and it is one of the most fundamental rules in all of biology. A pairs with T, G pairs with C, always, without exception, in any cell, in any organism, anywhere on Earth. If you know the sequence of one strand of DNA, you can always determine the sequence of the other strand with complete certainty simply by applying this rule: O with T, G with C. A quiet, reliable rule that the universe has decided to follow, and in following that rule, consistently, unfailingly, at every base pair, in every cell of every organism that has ever lived, the continuity of life is made possible. Fact 8. The bonds that hold the base pairs together, the bonds that form the rungs of the spiral staircase, are called hydrogen bonds. These are relatively gentle bonds. They are weaker than many other types of chemical bonds, and that gentleness turns out to be very important because it means the 2 strands of DNA can be carefully pulled apart when the cell needs to read or copy the genetic information, and then allowed to come back together again gently pulled apart, and gently brought back together, the molecule opens, like a very long, very thin book, and then closes again, over and over, throughout the life of the cell, a quiet, recurring action, at the heart of every living thing. Let's move, just a little more deeply, into the chemistry of DNA. We'll take each piece, one at a time, setting it down gently. Before we pick up the next, there is no need to hold all of it at once. The outside of each DNA strand, the long continuous rail of that spiral staircase, is made of two types of molecules, alternating in a regular pattern. One type is a sugar, the other is a phosphate group. Sugar, phosphate, sugar, phosphate, repeating over and over along the entire length of the strand. This sugar-phosphate pattern forms the structural backbone of the molecule, the framework that everything else hangs from. Fact 9. The sugar in DNA is called deoxyribose. It is a 5-carbon sugar, meaning that its central structure is built around a ring of 5 carbon atoms. The name, deoxy, tells us something specific. It means that this sugar is missing one oxygen atom, compared to a very similar sugar called ribose. Ribose is the sugar found in RNA, DNA's close molecular cousin. That single missing oxygen atom, that one small difference, contributes to making DNA more chemically stable than RNA, and therefore better suited for long-term storage of genetic information. Missing one oxygen atom, one atom out of the dozens that make up the deoxyribose molecule. And yet, that one difference has consequences that ripple outward, shaping the stability of the molecule, shaping how it is used by the cell, shaping the history of life itself. Fact 10. RNA stands for ribonucleic acid, while DNA tends to remain safely stored inside the nucleus. RNA is more of a working molecule, more mobile, more versatile. RNA carries copies of genetic instructions out of the nucleus and into the wider interior of the cell, where those instructions can be used. DNA holds the master library. RNA carries working copies out into the world. DNA holds the master library. RNA carries working copies. The two molecules complement each other, each doing what it is best suited to do. Together, they form the core of the cell's information system, and we can take a moment to notice how elegant that division of labor is. One molecule, stable, protected, stored. The other, mobile, active, temporary. Two molecules, working together, each one specialized, each one essential. Fact 11. The phosphate groups in the DNA backbone give the molecule a negative electrical charge. This charge runs along the entire length of the molecule. The electrical charge has several important consequences. It means that DNA repels other negatively charged molecules, keeping certain things away, and it means that positively charged proteins can bind to DNA quite readily, which is how the DNA is organized, packaged, and regulated inside the cell. Negative attracting positive, a steady, quiet electrical attraction, drawing the right proteins to the DNA, not a dramatic force, but a consistent one, working continuously, shaping the organization of the molecule in every cell of every living thing. Fact 12. The proteins that help package DNA inside the cell nucleus are called histones. Specifically, DNA wraps around small clusters of 8 histone proteins. The histone cluster, with the DNA wound around it, forms a structure called a nucleosome. A nucleosome looks, if you could somehow see it, a little like a small, round spool of thread with the DNA wound around it. Approximately 1.7 times. Many nucleosomes are strung along the DNA, one after another, creating a structure that looks like beads on a very long, very thin string. Beads on a string. It is a simple and useful image. Nucleosomes, the beads, strung along the DNA, the string, creating the first layer of organization, the first step in compacting that long molecule into something small enough to fit inside the nucleus. Fact 13. The string of nucleosome beads is then coiled into a thicker structure. That thicker structure is then looped. Those loops are then organized and compacted further, and then further still, layer upon layer of folding and packing, each one building on the last, until eventually the DNA is compressed into the dense, compact structure we call a chromosome. This process of hierarchical folding, fold upon fold, is what allows 2 meters of DNA to fit inside a nucleus that is only 5 to 10 micrometers across. Fold upon fold, each level of folding, a kind of quiet miracle of engineering, the body achieving, in every cell, a degree of compact organization that human engineers have never been able to replicate at this scale. And we can take a moment to notice, gently, that this extraordinary organization is happening right now in the nucleus of each of the trillions of cells in your body, without any effort on your part, without any awareness required, the body organizing itself quietly and continuously in the dark. Let's think quietly about where DNA lives. We've mentioned the nucleus several times already. Let's spend a few slow, peaceful minutes inside it. The nucleus is a small, roughly spherical compartment inside the cell. It sits somewhere near the center of the cell, though its exact position varies. The nucleus is surrounded by a membrane, not one layer, but two, a double membrane that creates a protected inner space. The double membrane is called the nuclear envelope, and it is the boundary between the nucleus, the inner room, where the DNA lives, and the rest of the cell. Fact 14. The nuclear envelope is not a solid, sealed wall. It is punctuated by many small openings, called nuclear pores. These pores are not simple holes. They are complex, protein structures that act as carefully controlled gateways, allowing specific molecules to pass in and out of the nucleus while maintaining the separation between the nucleus and the cytoplasm. Through these pores, RNA molecules carry genetic information outward. Through these pores, proteins and other molecules that the nucleus needs are imported inward. Controlled gateways, a selective border, the nucleus, protected but not sealed, open to what it needs, closed to what it doesn't. Fact 15. The nucleus of a human cell is somewhere between 5 and 10 micrometers in diameter. A micrometer is 1/1,000,000th of a meter, 1,000th of a millimeter. So the nucleus is extraordinarily small, invisible to the naked eye, and yet, within it, all of that DNA, 2 meters of it, is folded and organized into chromosomes, stored in a space that a grain of sand would dwarf, 5 to 10 micrometers. The nucleus, an entire library of biological instruction, smaller than the period at the end of this sentence, and inside it, the double helix, patient and folded, waiting. Fact 16. Within the nucleus, DNA is organized into distinct structures called chromosomes. In human cells, there are 46 chromosomes in each body cell, arranged in 23 pairs. Every pair consists of one chromosome, inherited from the mother, passed down through the egg cell, and one chromosome, inherited from the father, passed down through the sperm cell. So every cell in your body carries a quiet, combined record of both your parents, their genetic contributions. Side by side, a combined record, two parents represented in every cell, the cell carrying that history, without any effort, without any awareness, simply organizing it and maintaining it as it always has, as cells always have. Fact 17. The 46 human chromosomes vary considerably in size. The largest, chromosome 1, contains approximately 248 million base pairs of DNA. The smallest, chromosome 21, contains about 47 million base pairs. Even the smallest chromosome, in isolation, is an enormous repository of genetic information, millions of letters of biological instruction, carrying the instructions for dozens of genes, 47 million base pairs, the smallest chromosome. And yet, 47 million is still an immense number. Even the least of the chromosomes carries a vast library within it. Fact 18. The 23 pairs of human chromosomes consist of 22 pairs, called autosomes, chromosomes that are essentially the same in males and females, and one pair, called the sex chromosomes. Females typically carry 2 copies of the X chromosome, while males typically carry 1 X chromosome and 1 Y chromosome. The Y chromosome carries genes that direct male development. The X chromosome is considerably larger and carries far more genes, 2 X chromosomes. In females, 1 X and 1 Y. In males, the difference between the two most common biological sexes at the chromosomal level comes down to that 1 pair, while the other 22 pairs are shared. The common ground, as always, is larger than the difference. Let's now think carefully and slowly about what the sequence of bases in DNA actually means. We've established that DNA contains 4 letters, A, T, G, and C, arranged in a particular order along the molecule. But how does that sequence of letters carry information? How does a long string of 4 repeating letters become a set of instructions for building a living organism? The answer is something called the genetic code, and like so many things in biology, it is both elegantly simple and surprisingly deep. Let's take it very slowly. Fact 19. The genetic code works by reading the DNA sequence in units of 3 bases at a time. Every 3 consecutive bases along the sequence, every 3 letters work together to form a unit called a codon. Each codon specifies a particular amino acid, the building block of a protein. So the sequence of codons in a gene specifies the sequence of amino acids in the protein. That gene encodes 3 letters at a time: ADC, GJ, tag. Each triplet a small, quiet instruction, each codon saying, add this amino acid to the chain you are building. Add this one, then the next one, then the one after that. Fact 20. There are 4 possible bases, and each codon is 3 bases long. This means the total number of possible codons is 4 times 4 times 4, which equals 64. Of those 64 codons, 61 are used to specify amino acids. The remaining 3 codons serve as stop signals. They tell the protein-building machinery that the instructions for this particular protein have come to an end. 64 possible codons. 61 specifying amino acids. 3 acting as stops. A complete grammar in 3-letter words with punctuation. Fact 21. Because there are 64 codons, but only 20 amino acids used by living things. Most amino acids are specified by more than one codon. For example, the amino acid leucine can be specified by 6 different codons. This redundancy in the genetic code is not an accident. It has a protective quality. If a mutation changes one base in a codon, there is a reasonable chance the new codon still codes for the same amino acid, and the protein is unchanged. Redundancy with a protective quality. The code is forgiving. It has a gentleness built into its structure, a tolerance for small errors that allows life to carry on even in the face of occasional mistakes. Fact 22. The stop codons, the 3 codons that signal the end of a protein, are UA, UAG, and UGA. You'll notice that these are written with AU rather than AT. That's because the genetic code is usually described in terms of the RNA sequence, not the DNA sequence, since it is the messenger RNA that is actually read by the protein-building machinery. In RNA, uracil, U, replaces thymine, T, but it pairs with the same base, adenine, uracil, replacing thymine, a small substitution. In the working copy, the DNA uses T, the RNA uses U, but the information is the same, like a word, spelled slightly differently, in 2 dialects, but meaning exactly the same thing. And we can take a gentle moment to appreciate how much meaning is packed into 4 letters, and a simple rule about reading in triplets all of the genetic information in all living things encoded in a system this compact. Now let's follow the path from the DNA sequence to the finished protein. This journey has 2 main stages, and we'll follow each one slowly and gently. The first stage is called transcription. Fact 23. Transcription is the process by which the information stored in a gene's DNA sequence is copied into a molecule of messenger RNA or mRNA. During transcription, a large protein complex called RNA polymerase attaches to the DNA near the beginning of a gene and begins moving along the DNA strand, reading it base by base. As it reads, it builds a complementary strand of mRNA following the same base pairing rules we've already learned, but using uracil instead of thymine. RNA polymerase moving steadily along the DNA, reading, building, moving forward, like a patient reader moving through a very long text, copying it word by word into a new document. Fact 24. The region of DNA where RNA polymerase first attaches near the beginning of a gene is called the promoter. The promoter is a specific DNA sequence that signals this is where a gene begins, and this is where transcription should start. Different genes have different promoter sequences, and the promoter sequence influences how strongly and how often the gene is transcribed. The promoter, the beginning, the place where the machinery attaches and the reading begins. Every gene has one, quietly marking the start of its instructions. Fact 25. Once the mRNA strand has been assembled, it is released from the DNA template. In human cells, the newly made mRNA undergoes several modifications before it is ready to be used, including the addition of a protective cap at one end, a long tail of adenine bases at the other end, and the removal of non-coding sequences called introns. Once these modifications are complete, the processed mRNA is ready to be exported through the nuclear pores into the cytoplasm. A protective cap, a long tail, non-coding sequences removed, the mRNA carefully prepared and processed before it is sent out into the wider cell. The nucleus is thorough In its work, Fact 26, the non-coding sequences, removed from the mRNA during processing, are called introns, and the coding sequences that remain are called exons. The introns are spliced out, and the exons are joined together to form the continuous mRNA sequence that will be translated into protein. This process of splicing allows different proteins to be made from the same gene by including different combinations of exons, introns, and exons spliced out and joined together. The possibility of different proteins from the same gene, depending on which exons are included, the genome more flexible, more versatile than a simple one gene, one protein view, would suggest. The second stage of the journey from gene to protein is called translation. This is where the mRNA sequence, the working copy of the gene's instructions, is decoded and used to build a protein. Fact 27. Translation takes place at structures in the cell called ribosomes. A ribosome is a large, complex molecular machine made of RNA and protein. It has two subunits, a large one and a small one, that come together around the mRNA strand. When translation begins, the ribosome moves along the mRNA, reading each codon and facilitating the addition of the appropriate amino acid to the growing protein chain. A large subunit and a small one coming together around the mRNA, beginning to move. The ribosome is one of the most ancient molecular machines in all of biology. It appears in essentially every known living organism, and its basic structure has been conserved for billions of years. Fact 28. As the ribosome moves along the mRNA, the amino acids are brought to it by small molecules called transfer RNA or tRNA. Each tRNA molecule carries one specific amino acid and has a three-letter anticodon that matches, complements, the corresponding codon on the mRNA. When the right tRNA arrives, its anticodon pairs with the codon, and the amino acid it carries is added to the growing chain. Then the tRNA releases, drifts away, and picks up another amino acid to try again, a molecular delivery service. Each tRNA specific to one amino acid and one codon, arriving, delivering, releasing, returning, the process repeating over and over until the entire protein chain is complete. Fact 29. When the ribosome reaches a stop codon on the mRNA, translation ends. A protein called a release factor recognizes the stop codon and triggers the release of the completed protein chain from the ribosome. The ribosome then separates into its two subunits, and the mRNA is released, ready to be translated again by another ribosome. The stop codon reached, the protein released, the ribosome disassembled, and ready for the next round. The machinery, patient and renewable, never really stopping, always beginning again. Fact 30. Once released, the protein chain must fold into its correct three-dimensional shape. This folding is critically important because a protein's function depends entirely on its shape. The folding process begins even as the chain is still being built and is influenced by the sequence of amino acids, their chemical properties, causing some parts of the chain to attract each other and others to repel until the chain settles into its lowest energy, most stable conformation, folding, the chain, finding its shape, chemistry, driving the protein toward the form it was always going to take, given its sequence, every protein's shape, ultimately, a consequence of its gene, and we can take a quiet moment to appreciate the whole journey from DNA in the nucleus to mRNA messenger to ribosome to protein, from archive to working copy to product, from the library to the worker's hands. Every time a cell divides, it must first make an exact copy of all of its DNA so that each daughter cell receives a complete genome. This process is called DNA replication. Let's think about it slowly and carefully. Fact 31. DNA replication begins at specific locations on the chromosome called origins of replication. These are particular sequences of DNA that serve as starting points, attracting the machinery that will open up the double helix and begin the copying process. In bacteria, there is typically just one origin of replication per circular chromosome. In human cells, there are thousands of origins distributed across all the chromosomes, so that replication can begin simultaneously at many points, allowing the entire 3 billion base pair genome to be copied in a matter of hours. Thousands of starting points, replication spreading outward from each one simultaneously, like quiet ripples spreading from thousands of points across the surface of a still lake. The parallelism makes the enormous task manageable. Fact 32. At each origin of replication, a group of proteins attaches to the DNA and begins to separate the two strands of the double helix, opening up the molecule so that each strand can serve as a template for building a new, complementary strand. The region where the two strands are being separated is called the replication fork, and it moves along the chromosome as replication proceeds, opening the double helix. As it goes, the replication fork moving along the chromosome, opening the double helix. Ahead of it, like gently parting a curtain, revealing the single strands. Within, behind it, the new strands growing, closing the helix. Back up again. Fact 33. The main enzyme responsible for building new DNA strands during replication is called DNA polymerase. This large, complex molecular machine attaches to the single-stranded DNA template and moves along it, reading each base and adding the complementary base to the new strand being built. It follows the same rule we've been using all evening. A pairs with T, G pairs with C, DNA polymerase, moving along the template, reading, building, moving on. In human cells, it works at a pace of approximately 50 new bases per second, slow by some measures, but extraordinarily accurate. Fact 34. DNA replication requires a short starter sequence, called a primer, before the main copying can begin. The primer is a short stretch of RNA built by a separate enzyme, called primase. Primase attaches to the single-stranded DNA template and constructs the short RNA primer, giving DNA polymerase a place to attach and begin adding DNA bases. Once replication is complete, the RNA primers are removed and replaced with DNA by other enzymes, and the gaps are sealed. The primer, a small beginning, a few quiet letters of RNA placed at the start, giving the larger machinery a foothold from which to begin its work. Fact 35. Because DNA polymerase can only work in one direction, and because the two template strands of the double helix run in opposite directions, one new strand can be built smoothly and continuously in the direction of the moving replication fork. The other new strand, however, must be built in the opposite direction, in small fragments, one after another, each beginning with its own primer. These fragments are called Okazaki fragments, named after Reiji and Suniko Okazaki, the Japanese scientists who discovered them. in the 1960s, Okazaki fragments, small sections, built separately, and then joined together, the cell finding an elegant workaround for a geometric constraint, the final result, a complete, continuous strand, assembled from many short pieces, sewn neatly together. Fact 36. DNA replication is extraordinarily accurate. The overall error rate, accounting for the proofreading activity of DNA polymerase and the subsequent DNA repair systems, is approximately 1 mistake per billion base pairs copied. That means that when the cell copies its entire genome of 3 billion base pairs, it makes, on average, only about 3 errors, copying the entire text of 10,000 novels and making 3 spelling mistakes. That is the precision of DNA replication. 3 mistakes in 3 billion letters. The precision is almost too large to hold in the mind, and even those 3 mistakes will likely be found and corrected by the cell's repair machinery before they can cause any lasting effect. Even with the extraordinary accuracy of DNA replication, and even with the careful proofreading built into DNA polymerase, errors do sometimes slip through, and DNA can also be damaged by things in the environment, by certain chemicals, by ultraviolet light from the sun, by radiation. The cell has evolved many specialized systems for detecting and repairing these errors and this damage. Fact 37. Scientists estimate that each of your cells experiences thousands of DNA-damaging events every single day. These include errors that escape the proofreading of DNA polymerase, damage caused by reactive oxygen molecules generated during normal metabolism, and damage caused by ultraviolet light and other environmental factors. The vast majority of these events are repaired quickly and silently by the cell's repair systems, leaving no lasting change. Thousands of damaging events per cell per day, and thousands of repairs keeping pace. The genome constantly under a gentle assault, constantly being maintained, the balance tipping quietly, reliably, always toward maintenance and repair. Fact 38. One of the simplest repair mechanisms is called base excision repair. In this process, an enzyme called a DNA glycosylase recognizes and removes a single incorrect or damaged base from the DNA strand, leaving a small gap. Another enzyme then removes the surrounding section of the strand. The gap is filled in with the correct sequence, using the other strand as a template, and the new segment is sealed in place. The process is precise, targeted, and constant. Precise and constant. The cell, monitoring its own DNA, recognizing problems, fixing them, moving on. The genome, maintained through continuous, quiet vigilance. Fact 39. A more extensive repair mechanism, called nucleotide excision repair, handles larger areas of damage, particularly the kinds of damage caused by ultraviolet light. UV light can cause 2 adjacent pyrimidine bases, typically 2 thymine bases, to form a chemical bond with each other, creating a distortion in the double helix called a thymine dimer. The cell recognizes this distortion, cuts out a section of the strand containing the dimer, fills in the correct sequence, and seals the strand. Thymine dimers, the invisible consequence of ultraviolet light. A small, local distortion in the double helix, recognized, excised, and repaired. The cell, undoing, silently, the damage done by the sun. Fact 40. One of the most serious kinds of DNA damage is a double-strand break, a place where both strands of the double helix are broken at the same location. Double-strand breaks can be caused by ionizing radiation and by certain chemicals. The cell has specialized pathways for repairing these breaks, including one called homologous recombination, which uses the other copy of the chromosome as a template for an accurate repair. Using the other copy as a template, the redundancy of having two copies of each chromosome, one from each parent, proving its value. When one copy is damaged, the other can serve as a guide for repair. Let's think now about mutations, the changes in DNA sequence that do occasionally occur, despite all of the cell's proofreading and repair activity. Mutation is a word that can sound alarming, but the reality of mutation is much quieter and more ordinary than that word might suggest. A mutation is simply a change in the sequence of DNA. One base replaced by another, one or more bases inserted where they shouldn't be, one or more bases deleted, or a larger rearrangement. At its most basic, a mutation is a change in the text of the genome. Fact 41. The simplest type of mutation is called a point mutation, the change of a single base pair to a different one. If an AT base pair is replaced by a GC base pair, that is a point mutation, depending on where in the genome the change occurs. A point mutation might have no effect at all, a small effect, or occasionally a more significant effect on the function of the gene. It falls within one base pair, one letter in the text of the genome, changed most of the time, inconsequential, occasionally meaningful. Fact 42. Because the genetic code is redundant, because most amino acids are specified by more than one codon, many point mutations in coding regions result in no change to the amino acid sequence of the protein. These are called silent mutations or synonymous mutations. The letter of the DNA code changed, but the meaning of the codon did not. Silent, the change happened, but the consequence was nothing. A synonym, replacing a word. Fact 43. Mutations that do cause a change in the amino acid sequence of a protein are called missense mutations. Some missense mutations change the protein only slightly, with no significant effect on its function. Others change the protein in ways that affect its shape, and therefore its function. The consequences depend entirely on which amino acid changed and where in the protein the change occurred. The consequences, depending on context, the same kind of change, one amino acid replaced by another, can be harmless in one position and significant in another, depending on the role that amino acid plays in the protein's three-dimensional shape and function. Fact 44. Over long timescales and across many generations, the accumulation of mutations in a population's DNA provides the raw material for evolution. Mutations that happen to be beneficial, that improve an organism's ability to survive or reproduce in its environment, tend to be passed on more frequently. Mutations that are harmful tend to be lost. Mutations that are neutral drift through the population over time, becoming more or less common. By chance, the neutral mutations, drifting, rising, and falling in frequency, like leaves on a slow, wide river, neither helped nor hindered by natural selection, simply present, accumulating, recording the passage of time. Let's return now to chromosomes and spend some quiet time at their very ends, because the ends of chromosomes have a fascinating structure, and that structure plays a quiet but significant role in the life of the cell. Fact 45. The ends of each chromosome are capped by special structures called telomeres. The word comes from the Greek for end and part. Telomeres are not genes. They do not code for proteins. Instead, they consist of a simple, repetitive sequence of DNA. In humans, the 6-base sequence T, T, G, 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 repeated hundreds to thousands of times, that serves to protect the end of the chromosome from degradation. T, T, A, G, 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 repeated hundreds to thousands of times, a simple, rhythmic cap at each end of each chromosome, like the plastic tip on the end of a shoelace, keeping the chromosome's ends from fraying, from being recognized as broken DNA by the cell's repair machinery. Fact 46. Each time a cell divides, the telomeres shorten slightly. This happens because DNA polymerase cannot completely copy the very end of a linear chromosome. A small section at each end is lost during every round of replication. Over many cell divisions, the telomeres gradually become shorter and shorter until eventually they reach a critical minimum length, shortening with each division very slowly over a lifetime of cell divisions, the telomere patiently marking the passage of time in the life of the cell. Fact 47. When telomeres become too short, the cell can no longer divide safely. At that point, the cell may enter a state called senescence, a kind of stable retirement from active dividing, or it may undergo programmed cell death, a controlled process called apoptosis. Cellular senescence and the gradual accumulation of senescent cells in tissues are thought to be one of the factors underlying the aging of organisms. Senescence, retirement, the cell, after many divisions, simply stopping, not dying immediately, but no longer dividing, sitting quietly in the tissue, doing some of its functions, but no longer contributing new cells. Fact 48. An enzyme called telomerase can rebuild telomeres. It adds new tag repeats to the ends of chromosomes, restoring their length. Telomerase is active in certain types of cells that need to divide many times without limit, including stem cells and the cells that give rise to eggs and sperm. In most other body cells, telomerase is present in very small amounts or is largely inactive, rebuilding the telomere, adding back what would otherwise be lost. The enzyme of renewal, available to some cells, but not most, the body's way of protecting its most important dividing cells while allowing others to age naturally. We have been using the word gene throughout this evening. Let's now take a few quiet minutes to really settle on what a gene is, simply and clearly. Fact 49. A gene is a segment of DNA that carries the instructions for making a specific product, usually a protein, or sometimes a functional RNA molecule that plays a role in the cell without being translated into protein. The genome is not one continuous, undifferentiated message. It is divided into many thousands of distinct sections, genes, each one dedicated to a particular purpose. Distinct sections, each one dedicated, each one a unit of instruction, bounded by its promoter and its stop codon, carrying within it the information for one specific product. Fact 50. The human genome contains somewhere between 20,000 and 25,000 protein coding genes. Before the Human Genome Project gave us the actual number, estimates had ranged up to 100,000 or more. The real number, around 20,000, came as a surprise to many scientists. It is not so many more genes than a simple nematode worm, which has approximately 2,500. The difference between a worm and a human lies not so much in the number of genes as in how those genes are regulated, combined, and expressed. Not so many more than a worm. The complexity of a human being arising not primarily from gene number, but from the elaborate regulation and interaction of a modest number of genes, the elegance of the system, more than the size of the parts. Fact 51. Protein coding genes make up only a small fraction of the total genome, approximately 1.5%. The remaining 98.5% was once dismissed by scientists as junk DNA, a term suggesting it was non-functional. We now know that much of this non-coding DNA plays important regulatory roles, helping to control when, where, and how much each gene is expressed. The genome is far from a simple collection of genes. It is a rich and complex regulatory landscape. A rich and complex regulatory landscape. Not junk, but a system of switches and signals controlling the activity of the genes themselves. Let's now move gently into a topic that adds a beautiful and subtle additional layer to everything we've been learning. This is epigenetics. The word epigenetics comes from the Greek for upon genetics or above genetics. It refers to changes in the activity of genes, changes in how genes are expressed that do not involve any change in the underlying DNA sequence. The letters of the genetic code remain exactly the same, but something about how those letters are read can change. Fact 52. One of the most important epigenetic mechanisms is DNA methylation. During DNA methylation, a small chemical group, called a methyl group, consisting of one carbon atom and 3 hydrogen atoms, is added to a cytosine base in the DNA. Typically, this happens at cytosines that are followed by guanines when certain regions of the genome are heavily methylated. The genes in those regions tend to be silenced, switched off, no longer actively transcribed. A methyl group placed on a cytosine, one small chemical addition, and the gene nearby may go quiet, its instructions no longer being read. The gene is still there, every letter still in place, but a small molecular tag has said, not this one, not here, not now. Fact 53. Methylation patterns vary between different cell types. Liver cells and brain cells contain the same DNA, but have different methylation patterns. Different genes are silenced in each cell type, creating the different patterns of gene expression that make a liver cell behave like a liver cell and a brain cell behave like a brain cell. The DNA sequence is the same. The epigenetic annotation differs. The same sequence, different annotations. The epigenome, as a layer of information placed on top of the genome, varying from cell to cell, helping each cell type express the right set of genes for its particular role. Fact 54. Environmental factors can influence DNA methylation patterns. Diet, stress, exercise, sleep, and exposure to certain chemicals have all been found in various studies to affect methylation at specific genomic locations. This is one pathway by which the external environment can influence gene expression, not by changing the sequence of DNA, but by changing the chemical tags placed on it, the external environment reaching inward, leaving a chemical trace. The genome, in quiet conversation with the world around it. Fact 55. Histone modifications are another important epigenetic mechanism. The histone proteins, around which DNA is wound, can be chemically modified in many ways: methylated, acetylated, phosphorylated, and each of these modifications influences how tightly the DNA is packaged around the histone. Tightly packaged DNA is generally less accessible to the transcription machinery, and therefore less actively expressed. Loosely packaged DNA is more accessible and tends to be more actively expressed. Tightly wound and loosely wound, open and closed, the physical arrangement of the DNA around its histone spools, shaping which genes are readable and which are not. Fact 56. The sum of all epigenetic modifications across the genome, all the methylation marks and histone modifications, and other regulatory annotations, is sometimes called the epigenome. Every cell has both a genome, the DNA sequence, and an epigenome, the pattern of modifications placed on that sequence. The epigenome varies between cell types, and it can change over the course of a person's lifetime. The epigenome changing over a lifetime. The genome stable, with the exception of mutations. 2 layers of information, both important, one fixed and one dynamic. Let's step back and think about DNA in the wider context of all living things. We have been focusing mainly on human DNA, but DNA is universal. Every known living organism uses it. Fact 57. The genetic code is nearly universal across all known life. The same codon that specifies a particular amino acid in a human cell specifies that same amino acid in a bacterium, in a fungus, in a plant, in a fish, with only very minor exceptions, mostly in mitochondria, and in a small number of unusual organisms. The genetic code is the same everywhere. This universality is one of the most compelling pieces of evidence that all life on Earth descends from a single, common ancestor. The same code everywhere, all of life connected by a shared molecular language, so ancient and so fundamental that it has not changed in billions of years of evolution. Fact 58. Bacteria have DNA, but their DNA is organized very differently from DNA in human cells. Bacterial DNA is not enclosed in a nucleus. Instead, it typically exists as a single, circular chromosome, floating in the cell's cytoplasm. Many bacteria also carry smaller rings of DNA, called plasmids, which can be exchanged between bacterial cells, a form of genetic sharing that is quite unlike anything in human biology. Circular, no nucleus, plasmids, exchangeable between cells. The bacterial world, doing things differently, doing them effectively, having done them for billions of years before the first plant or animal appeared. Fact 59. The size of a genome does not necessarily correlate with the complexity of the organism. The human genome contains approximately 3.2 billion base pairs. The genome of the Paris japonica plant, a flowering plant that grows in Japan, contains approximately 150 billion base pairs. That is about 50 times more DNA than the human genome. The relationship between genome size and organismal complexity is complex and not fully understood. 150 billion base pairs in a flowering plant, 50 times more than in a human. The genome size and the complexity of the organism, not always telling the same story. Fact 60. Many plants have multiple, complete sets of chromosomes, a condition called polyploidy. A diploid organism, like a human, has 2 sets of chromosomes. A tetraploid has 4 sets. A hexaploid has 6. Many of our important crop plants are polyploid. Wheat is hexaploid, for example, having 6 sets of chromosomes. Polyploidy is common in plants and can arise when cell division goes slightly wrong during the formation of reproductive cells. Hexaploid wheat, 6 sets of chromosomes, 42 chromosomes in each cell, compared to 46 in humans. The genomes of our food plants, carrying a history of polyploidy events, of ancient doublings and redoublings. Let's now move into something that is quietly wonderful: the ability of DNA to survive, in some circumstances, for thousands or even millions of years, allowing scientists to recover and read the genetic code of organisms that lived long before any human civilization existed. Fact 61. Ancient DNA is genetic material recovered from specimens that are hundreds, thousands, or even millions of years old. Cold, dry conditions slow the chemical degradation of DNA and can preserve it for remarkable lengths of time. The best conditions for preserving ancient DNA are found in permafrost, the permanently frozen ground of the Arctic regions, and in caves with stable, cold temperatures. Cold, dry, dark, the conditions that slow the chemistry of decay, holding the molecular structure intact across vast spans of time. The ice and darkness acting as a kind of archive. Fact 62. The field of ancient DNA research has grown dramatically since the first ancient DNA was successfully recovered in the 1980s. Early attempts worked with DNA from preserved museum specimens and ancient remains. As sequencing technology improved and methods for dealing with the highly fragmented, chemically damaged DNA in ancient specimens advanced, the field expanded rapidly, making it possible to recover genetic information from older and older specimens. The 1980s, just 4 decades ago, and already the field has advanced to the point where scientists can read the genome of a creature that lived 1 million years ago. Fact 63. In 2010, a landmark paper described the sequencing of the Neanderthal genome using DNA recovered from bone fragments found in a cave in Vindija, Croatia. The bones were approximately 40,000 years old. Comparing the Neanderthal genome to the genomes of living humans revealed that Neanderthals and anatomically modern humans interbred at some point during the period when the two populations coexisted in Europe and Western Asia. Interbred Neanderthals and modern humans. The evidence written into the genome of every person of non-African descent who typically carries between 1 and 4% Neanderthal DNA. Fact 64. Denisovans are another group of ancient humans known almost entirely through their DNA. The physical evidence, a handful of bone and tooth fragments from a cave in Siberia, is fragmentary, but the DNA from those fragments has been enough to reveal that Denisovans were genetically distinct from both Neanderthals and modern humans, that they interbred with both, and that their genetic legacy persists today in the genomes of some present-day populations, particularly in Southeast Asia and the Pacific, known through their DNA, the Denisovans, a people whose entire existence we would be unaware of were it not for the information preserved in a few fragments of bone in a Siberian cave. Fact 65. In 2022, scientists published the analysis of DNA from a woolly mammoth bone fragment recovered from Siberian permafrost. The bone was dated to more than 1 million years old, making it the oldest DNA ever successfully analyzed from a complex, multicellular organism. The preservation of the DNA over such an immense timescale was made possible by the consistently cold conditions of the Siberian permafrost. 1 million years. The number is difficult to hold. Modern humans have existed for only about 300,000 years. The woolly mammoths, whose DNA was analyzed, lived before our species existed, and yet the molecule endured. Let's pause and spend a few quiet minutes thinking about the Human Genome Project, the enormous scientific effort that gave us the first complete picture of the human genome. Fact 66. The Human Genome Project was an international scientific collaboration involving researchers from the United States, the United Kingdom, France, Germany, Japan, and China, among other countries. It was formally launched in 1990 and had the ambitious goal of determining the complete sequence of base pairs in the human genome, all 3 billion of them, and of identifying and mapping all human genes. An international collaboration, 6 countries, thousands of scientists, one shared an enormous goal. The project was, in its organization and its ambition, one of the largest coordinated scientific efforts in history. Fact 67. The Human Genome Project was completed ahead of schedule and under budget. A working draft of the genome, covering about 90% of the sequence, was announced in 2000. A more complete and accurate version was published in 2003. The total cost of the project was approximately $2.7 billion US dollars. Under budget, ahead of schedule, $2.7 billion. A very large number, but the value of what was produced has been immeasurable. Fact 68. One of the most striking things revealed by the Human Genome Project was how much of the genome does not consist of protein-coding genes. Scientists had assumed that a large proportion of the genome would consist of functional protein-coding sequences. The actual proportion, about 1.5%, was much smaller than expected. The rest of the genome, the vast majority, consists of regulatory sequences, repetitive elements, the traces of ancient transposons, and regions of uncertain or poorly understood function. 1.5%. The protein-coding genes, a small island in a vast ocean of other sequences. The ocean, not empty, but complex, regulatory, historical, full of signals we're still learning to read. Fact 69. Since the completion of the Human Genome Project, the cost of sequencing a human genome has fallen dramatically. In 2003, sequencing a single human genome cost approximately $2.7 billion. By 2023, the cost had fallen to below $1,000. This extraordinary reduction in cost has made genome sequencing a practical, clinical tool, and it has enabled research at scales that would have been unimaginable in 1990, from $2.7 billion to less than $1,000 in 2 decades, the most dramatic cost reduction in the history of any technology. Perhaps the genome going from an almost incomprehensibly expensive project to something routinely available. Let's think now about how our understanding of DNA has changed medicine and how it continues to do so. Fact 70. Genetic diseases. Diseases caused by mutations in specific genes are numerous and varied. Some are caused by mutations in a single gene, and their inheritance follows relatively predictable patterns. Others are influenced by variations in multiple genes interacting with each other and with environmental factors. The identification of the specific genetic basis of many diseases has transformed our ability to diagnose, understand, and sometimes treat them. Single-gene diseases and complex, multi-gene ones, simple inheritance, and complex interactions. The spectrum of genetic disease, as broad and varied as life itself. Fact 71. Cystic fibrosis is caused by mutations in a single gene called CFTR, which encodes a protein that forms a channel, allowing chloride ions to pass through the membranes of cells. When this channel doesn't work correctly, secretions in the lungs, digestive system, and other organs become abnormally thick and sticky. Over 1,700 different mutations in the CFTR gene have been identified, each affecting the protein in a slightly different way. Over 1,700 different mutations in one gene, each one telling a slightly different story about how the protein can fail. The diversity of mutations, a testament to the many ways in which a gene can be changed by the patient. Slow accumulation of chance events over generations. Fact 72. Sickle cell disease is caused by a single point mutation, one base pair, changed in the gene for the beta chain of hemoglobin. This one-letter change causes the hemoglobin protein to behave abnormally under low oxygen conditions, causing it to form long fibers that distort red blood cells into a sickle shape. The sickled cells can block small blood vessels and cause pain, anemia, and organ damage. One letter changed in the entire genome, one base pair altered, and the consequence cascading outward from that one molecular change to the protein, to the cell, to the tissue, to the patient. Fact 73. Newborn screening programs test infants' blood shortly after birth for a panel of genetic conditions that, if detected early, can be effectively managed. In many countries, newborns are screened for dozens of conditions. The goal is to identify those who might benefit from early intervention before symptoms appear, because for many genetic conditions, early treatment can prevent serious harm. Early intervention. The quiet, practical application of genetic knowledge. A few drops of blood analyzed for molecular markers, translating into early treatment and prevented harm. Fact 74. The BRCA1 and BRCA2 genes encode proteins that help repair damaged DNA. When these proteins function normally, they help protect the genome from the kind of damage that can lead to cancer. When the BRCA1 or BRCA2 genes carry certain mutations, this repair function is impaired, and the risk of certain cancers, particularly breast and ovarian cancer, is significantly elevated. Identifying these mutations allows individuals to make informed decisions about monitoring and prevention. DNA repair genes connected to cancer risk. The link between the molecular machinery of DNA repair and the development of cancer, one of the most important insights of modern molecular biology. Fact 75. CRISPR stands for clustered regularly interspaced short palindromic repeats. It is a natural system found in bacteria where it functions as a form of adaptive immunity. When a bacterium survives an attack by a virus, it can store small fragments of the virus's DNA in its own genome in the CRISPR region, so that if the same virus attacks again, the bacterium can recognize it and mount a targeted defense. An immune memory stored in DNA, the bacterium learning from its encounters with viruses, keeping a molecular record of what it has faced, using that record to defend itself in the future. Fact 76. Scientists recognized that the CRISPR system, particularly a protein called Cas9, which acts as the cutting enzyme in the bacterial defense system, could be adapted into a precise gene editing tool for use in other organisms by designing a short guide RNA that matches the target DNA sequence. Researchers can direct the Cas9 protein to any specific location in any genome and make a precise cut any location in any genome. The guide RNA, a molecular address label, Cas9, following that address, finding the location among billions of base pairs, and cutting. Fact 77. Jennifer Doudna of the University of California, Berkeley, and Emmanuelle Charpentier, then at the Helmholtz Center for Infection Research in Germany, collaborated to demonstrate the potential of CRISPR-Cas9 as a gene editing tool. In 2012, their paper describing the technology was published in the journal Science. In 2020, they were jointly awarded the Nobel Prize in Chemistry for this work. The Nobel Prize in Chemistry 2020, two scientists, a collaboration, a single paper, a technology that has transformed biology in the decade since its publication. Fact 78. CRISPR-Cas9 has been applied in many different areas of biology. In basic research, it is used to study gene function by allowing researchers to disable specific genes in cell cultures or model organisms and observe the effects. In medicine, it is being developed as a treatment for genetic diseases. In agriculture, it is being used to develop crop plants with improved resistance to disease, drought, and pests. Many applications: research, medicine, agriculture, the tool, versatile and precise, spreading across the biological sciences. Fact 79. In late 2023, the first CRISPR-based therapy, a treatment for sickle cell disease and beta thalassemia, two inherited blood disorders, was approved by regulatory authorities in the United Kingdom and the United States. The therapy works by using CRISPR to edit the patient's own blood stem cells, activating a gene that produces a form of hemoglobin normally found only in fetuses. This fetal hemoglobin functions normally, even in patients with mutations in their adult hemoglobin gene. Fetal hemoglobin reactivated in adult cells, a molecular workaround, switching on a gene that was silenced during development. The first approved CRISPR therapy, a milestone in the long path from scientific discovery to clinical medicine. Fact 80. Forensic DNA analysis is based on the fact that the genomes of different individuals differ at many specific locations. At these locations, called polymorphic sites, different people carry different versions of the sequence. By comparing the patterns of variation at a sufficient number of these sites, a genetic profile can be constructed that is statistically unique to one person, with the exception of identical twins who carry the same genome. Statistically unique, not a certainty, but a probability so high as to be effectively definitive, the genome's natural variation turned into a tool for identification. Fact 81. Short tandem repeats, or STRs, are one of the most commonly used types of genetic variation. In forensic profiling, at an STR locus, a short sequence, perhaps 4 or 5 bases long, is repeated many times in a row. Different individuals typically have different numbers of repeats at any given STR location. By analyzing multiple STR locations simultaneously, forensic scientists can generate a highly discriminating genetic profile, different numbers of repeats at multiple locations. The combination, specific enough to identify individuals with high confidence from very small biological samples. Fact 82. DNA can be recovered from trace biological material, a single hair, a few skin cells, a small drop of saliva or blood, the residue left by a touch. Modern analytical techniques are sensitive enough to generate a DNA profile from extremely small samples, making forensic DNA analysis applicable in situations where other types of physical evidence might be absent. Extremely small samples, the sensitivity of modern molecular biology applied to criminal investigation. Fact 83. The Innocence Project is an organization founded in 1992 that uses DNA evidence to help exonerate people who have been wrongfully convicted of crimes. Since its founding, the Innocence Project and affiliated organizations around the world have helped free more than 375 people in the United States alone, using DNA evidence that demonstrated their innocence. Many of these individuals had served decades in prison, more than 375 people freed by the evidence in their DNA. The molecule, in this application, acting as a quiet and reliable witness. Fact 84. Life on Earth is estimated to have begun approximately 3.5 to 4 billion years ago. The earliest direct evidence comes from fossilized stromatolites, layered structures built by communities of photosynthesizing cyanobacteria found in rock formations in Western Australia, dated to approximately 3.5 billion years ago. Living stromatolites can still be found in a few coastal locations around the world, most famously in Shark Bay, Western Australia, 3.5 billion years. Stromatolites still living in a few quiet corners of the world, the same basic biological structures persisting across almost unimaginable spans of time. Fact 85. The transition from non-living chemistry to living chemistry, the origin of life, remains one of the deepest open questions in all of science, but significant progress has been made in understanding the conditions that might have allowed the first self-replicating molecules to arise. The early Earth's oceans and volcanic systems provided a rich chemical environment with many of the building blocks of biological molecules present. An open question, a deep one, the origin of life. The moment when chemistry became biology remains fascinating, precisely because It is not yet fully understood. Fact 86. The RNA world hypothesis proposes that, before DNA and proteins, an earlier stage of life was based entirely on RNA. RNA molecules are capable of both storing genetic information, like DNA, and catalyzing chemical reactions, like proteins. This dual capability makes RNA a plausible candidate for the first self-replicating molecule capable of making copies of itself and of catalyzing the reactions necessary to do so. RNA doing two jobs at once: storage and catalysis. The hypothesis is supported by the discovery that the catalytic center of the ribosome, the molecular machine that builds all proteins, is made of RNA, not protein. The ribosome's core is an RNA enzyme, a ribozyme, suggesting that RNA catalysis came before protein catalysis. Fact 87. Over time, in the RNA world scenario, DNA would have evolved as a more stable alternative for genetic storage, while proteins would have evolved as more versatile and effective catalysts. RNA would have been gradually displaced from both roles, relegated to its current function as the intermediary between DNA and protein. The central position of RNA in modern cells as mRNA, tRNA, rRNA, and many regulatory RNAs may be a fossil of this ancient RNA world, a fossil of the RNA world, the RNA molecules in your cells performing functions that once fell entirely to RNA before DNA and protein took over. Ancient systems, still running, still essential. Fact 88. The atoms that make up DNA, carbon, hydrogen, nitrogen, oxygen, and phosphorus, were created through cosmic processes that began long before the Earth existed. Hydrogen, the most abundant element in the universe, was created in the first few minutes after the Big Bang, approximately 13.8 billion years ago. The heavier elements, carbon, nitrogen, oxygen, phosphorus, were produced later, inside stars. The Big Bang, 13.8 billion years ago. The hydrogen in the water molecules around you, in the hydrogen bonds of your DNA, created in the first moments of the universe's existence. Fact 89. Carbon, nitrogen, and oxygen are produced in the cores of massive stars through nuclear fusion. When a star runs out of fuel, it explodes in a supernova, a catastrophic stellar explosion that scatters the elements produced during the star's life across space. The carbon, nitrogen, and oxygen in your body were produced in the cores of ancient stars and dispersed into space by stellar explosions, dispersed into space, drifting for billions of years in the interstellar medium, gathered, eventually, by gravity, into the cloud of gas and dust that became our solar system, incorporated into the young Earth, dissolved in the early oceans, assembled by chemistry into the first biological molecules. Fact 90. The process by which early atoms assembled into the building blocks of life, amino acids, nucleotides, sugars, is an active area of scientific research. Experiments going back to the 1950s have shown that many of these building blocks can be formed spontaneously under conditions that might have existed on the early Earth. Amino acids, for example, can be produced by passing an electric spark through a mixture of simple gases, water, methane, ammonia, hydrogen, mimicking the conditions that might have prevailed in the early atmosphere, spontaneously formed, the building blocks of life, emerging from simple chemistry, under the right conditions. No special direction required, just chemistry, time, and the right environment. Fact 91. Mitochondria, the energy-producing organelles of the cell, have their own DNA, separate from the nuclear genome. Human mitochondrial DNA is a small, circular molecule of 16,569 base pairs, containing genes, 413 proteins, 22 transfer RNAs, and 2 ribosomal RNAs, all 13 proteins, encoded in the mitochondrial genome, are components of the electron transport chain, the molecular machinery that produces most of the cell's energy, 16,569 base pairs. Small, by comparison, to the nuclear genome, but essential. The proteins encoded in mitochondrial DNA are central to the energy-generating machinery on which all cellular activity depends. Fact 92. The endosymbiotic theory proposes that mitochondria evolved from free-living bacteria that were engulfed by a larger cell approximately 1.5 to 2 billion years ago. Rather than being digested, the bacterium and the host cell entered into a mutually beneficial relationship. The bacterium providing energy, the host cell providing protection and nutrients. Over time, the bacterium lost its ability to live independently, and most of its genes were transferred to the host cell's nucleus. A partnership struck 2 billion years ago that has persisted in every animal, every plant, every fungus, ever since. The mitochondrion, once free, now an integral part of every cell, carrying still its small circular chromosome, a molecular echo of its bacterial origins. Fact 93. Mitochondrial DNA is inherited almost exclusively through the maternal line. The mitochondria in a fertilized egg come almost entirely from the egg cell. The sperm contributes only its nucleus. As a result, you inherited your mitochondrial DNA from your mother, who inherited hers from her mother, who inherited hers from her mother, and so on, back through an unbroken maternal line, stretching for thousands of generations. An unbroken maternal line, thousands of generations. Your mitochondrial DNA, a thread connecting you to a long sequence of mothers, each one inheriting and passing on the same small, circular molecule. Fact 94. Because mitochondrial DNA accumulates mutations at a relatively steady rate, over time, it can be used as a molecular clock, a way of estimating how long ago two lineages diverged from a common ancestor. By comparing the mitochondrial DNA of living humans from different populations, scientists have estimated that all living humans share a most recent, common matrilineal ancestor, sometimes called mitochondrial Eve, who lived approximately 150,000 to 200,000 years ago. Most likely, in Africa, mitochondrial Eve, not the only woman alive at the time, but the one whose mitochondrial lineage has survived, unbroken, to the present day, a thread 150,000 years long, connecting every living human being through their mitochondria. Fact 95. Barbara McClintock was an American geneticist who spent decades studying the genetics of maize, corn, at the Cold Spring Harbor Laboratory in New York. In the 1940s and 1950s, she discovered that certain genetic elements could move around within the genome, inserting themselves in new locations, sometimes disrupting gene function, sometimes creating new patterns of gene regulation. She called these mobile elements transposons. mobile elements, moving around, within the genome. The idea, radical at the time, that genes could change their location, that the genome was not a fixed map, but a more dynamic landscape. Fact 96. McClintock's discovery was not immediately accepted by the scientific community. Her work was dismissed or ignored by many of her contemporaries who could not fit the idea of mobile genetic elements into their existing understanding of genetics. She continued her work quietly and independently, publishing her findings, even as they went, largely unrecognized, for many years, quietly and independently, continuing the work. The story of Barbara McClintock is in many ways the story of a scientist ahead of her time, persisting in the face of skepticism, confident in the evidence. Fact 97. Barbara McClintock was awarded the Nobel Prize in Physiology or Medicine in 1983, more than 3 decades after her initial discovery. By that time, transposons had been found in organisms, from bacteria to humans, and their importance for genome evolution, gene regulation, and the generation of genetic diversity was fully appreciated. McClintock was 81 years old when she received the prize. 81 years old, decades after the discovery, the recognition, finally, for work that had transformed biology. Her patience, her persistence, and her quiet confidence in her own observations vindicated, at last. Fact 98. Transposons make up approximately 45% of the human genome. Nearly half of your DNA consists of transposons, or the remnants of ancient transposon activity, sequences that have inserted themselves into the genome over the course of evolution, accumulated mutations over millions of years, and been largely silenced by epigenetic mechanisms. They're a record of the genome's history, embedded within it, nearly half the genome as a layered historical document, 45% of it the accumulated traces of mobile elements stretching back into deep evolutionary time. Fact 99. Some transposons, called lines, long interspersed nuclear elements, can still move within the human genome, particularly in certain cell types, including neurons. The movement of lines in brain cells is thought to contribute to the genetic diversity that exists between different cells within a single brain and may play a role in brain development and function. The mobile genome, still active, still contributing to the diversity of the organism, still active, still moving. The genome, even now, not entirely static, evolving quietly, internally, even within the body of a single individual. Fact 100. Every human being who has ever lived has carried within their cells the same fundamental molecule, DNA, with its double helix, its 4 bases, its elegant base pairing rules, its genetic code, from the earliest, anatomically modern humans who walked across ancient African landscapes more than 300,000 years ago, to every person alive on Earth today. The molecule is the same. The 4 letters are the same. The rule, A with T, G with C, is the same. The language of life, unchanged in its essentials, passed from generation to generation, across all the centuries and all the generations of human history. The same molecule, the same language, passed on, every person carrying it, every person, a link in a chain that stretches back before humanity, before animals, before complex life, to the very first cells that arose in the ancient oceans of a young Earth. We have now visited all 100 of our quiet scientific facts about DNA, and together we have covered a great and gentle distance from the shape of the molecule to the code it carries, to the machinery of the cell, to the history of life, to the stars that made our atoms. Now, it is time to begin slowing down, not stopping suddenly, just slowing, the way a river slows as it approaches the sea, the water, still moving, still carrying everything. It is gathered, but moving more gently, more broadly, spreading into calm. Let's take a long, slow breath together, in through the nose, and out through the mouth. Let the body settle a little further into whatever it's resting on. Let the weight of the body, the comfortable heaviness of it, be a welcome feeling. The DNA in your cells has been doing its patient work all evening as you've been listening. It was doing it before you started listening. It will continue doing it after you fall asleep. The ribosomes, assembling proteins, the repair proteins, monitoring the genome, the transcription factors, binding to promoters, the histones, holding the DNA in their quiet coils, all of it continuing without any need for your attention, without any effort on your part. The body knows what to do, and you can simply rest in the quieter and more spacious part of the evening. Let's return to the very simple things, the first things we learned, the things that have been with us all along. DNA is a molecule. It is shaped like a double helix, two strands winding around each other like a long and patient spiral. The strands are held together by base pairs, adenine with thymine, guanine with cytosine, O with T, G with C, always, without exception. The sequence of those base pairs, the order in which A, T, G, and C appear along the strand, is the genetic code. That code is read in triplets, 3 letters at a time, each triplet specifying an amino acid, amino acids assembled into proteins, proteins doing the work of the cell. The DNA is wound around histone proteins, organized into nucleosomes, folded into chromosomes, held inside the nucleus, inside the cell, inside you. Let's let those ideas rest very gently, not holding them, not memorizing them, just letting them be there, like furniture in a familiar room, present, quiet, known, O with T, G with C, the rule, followed, in every cell, in every organism, on every continent, in every sea, at every moment, since the first living cells arose in the ancient oceans of a young earth, O with T, G with C, the universe, following its own quiet rules. Let's return one more time to the thought that brought us closest to the very large. The atoms in your DNA were made in stars, carbon forged in the nuclear furnaces of ancient massive stars, nitrogen created there too, oxygen, phosphorus produced in the final moments of a dying star's life, in a furious last burst of nuclear burning, before the explosion that would scatter it all across space. Hydrogen, created in the first minutes after the Big Bang, 13.8 billion years ago. Those atoms, drifting through interstellar space for billions of years, gathered by gravity into a new cloud of gas and dust. That cloud collapsing slowly, forming a new star, our sun, and around it, a disk of material from which the planets condensed, the Earth assembling itself from that ancient stellar material, the oceans gathering, the early atmosphere forming, chemistry happening in the oceans and on the shores, slowly patiently, over hundreds of millions of years. And then, at some point, perhaps in a warm, shallow pool, perhaps in the hot water, around a deep-sea vent, perhaps somewhere else, entirely, the first molecules, capable of copying themselves, the first hint of heredity, the first flicker of something we might one day call life. And from that flicker, from that first quiet act of molecular self-replication, everything else followed. Every organism that has ever lived, every bacterium, every plant, every animal, every human being, including you, including the atoms of your DNA, which were inside stars, long before they were inside you. Let that thought rest, very gently. It is not something to hold tightly. It is something to let settle, like starlight settling on a still lake at night, softly, from a great distance, quietly arriving. It is late now, or early, the hours that don't quite belong to either day. Your body is at rest. Your breathing is slow. The room around you is still, and inside you, in the nucleus of each of your cells, the DNA is folded and quiet, tag, repeated, at the ends of each chromosome, capping them, protecting them, the histones, holding the chromatin in its organized coils, the nuclear pores, maintaining their selective watch, the repair proteins, monitoring the genome for damage, finding almost none, the ribosomes, building proteins, slowly, steadily, one amino acid at a time, the sequence, encoded in the DNA, expressed through the mRNA, translated by the ribosome into the protein chain, folded into its correct shape, released into the cell to do its work. All of this happening right now in trillions of cells, simultaneously, without any awareness on your part, without any effort, the body doing what bodies have always done in the quiet hours, in the dark. You don't need to watch it. You don't need to think about it. The cell has been doing this for longer than consciousness has existed. It doesn't need supervision. You just need to breathe in and out, the breath arriving and leaving as it always has, as it will continue to. The words can come more slowly now, with more space between them, like the way the mind begins to slow when sleep is close. DNA, a double helix, two strands winding, O with T, G with C, 320 million turns of the helix in the nucleus of each cell, each turn patient and regular, each turn the same, O with T, G with C, over and over, 320 million times, folded and still, in the quiet interior of the nucleus, in the quiet interior of the cell, in the quiet interior of you. Think of a single cell, somewhere in your body, perhaps a cell in the skin of your hand, or a cell in the muscle of your shoulder, warm and relaxed, or a cell deep in the tissue of your heart, beating steadily, without any thought from you. Inside that cell, inside its nucleus, is a long wound molecule, 2 meters of it, folded many thousands of times, organized into 46 chromosomes, wrapped around histone proteins, held inside a space, a few micrometers across. The cell does not know that you are resting. It does not know that it is late. It knows only what cells have always known, the ancient chemistry of life, encoded in the DNA, expressed through the quiet machinery of proteins and RNA, and it continues, as it always does, as it always has. Your body right now is building proteins, is repairing DNA, is maintaining the integrity of the genome, is producing energy in the mitochondria, is continuing the 10,000 chemical reactions that constitute being alive, all of it quiet, all of it invisible, all of it happening without you. Think, if you wish, of the long chain of generations that connects you to the past, your parents, who carried in their cells the DNA that was combined to make yours, their parents before them, and their parents, generation by generation, backward into time, 100 generations ago, 1,000 generations ago, 10,000 generations ago, 20,000 generations ago, and at each point, in that chain, in each body, in each cell, the same molecule, the same 4 letters, the same double helix, the same patient rule, A with T, G with C, followed without exception, passed from parent to child. You are the current carrier of a very old message, a message that has passed through more generations than any human mind can comfortably count, and it will pass on into more generations still, into a future as long and patient as the past. There is something quietly grounding in that, not dramatic, just steady, the sense of being part of something very old and very continuous, a molecule, a code, a chain of life, stretching from the ancient past into the unknown future. Very gentle now, the DNA molecule is there in the nucleus, patient, folded, at rest, A with T, G with C, the helix turning, slow and patient, each turn the same, each base pair, following the rule, the histones holding the DNA, the nuclear envelope, maintaining its quiet boundary, the telomeres, capping the ends, everything in its place, everything organized, everything at rest. And you're at rest too, heavy and warm, breathing slowly, the body knowing what it knows, the cell knowing what it knows, the molecule knowing what it has always known. The night is very deep now. Let the body be heavy. Let the mind be soft. Let the thoughts come and go. A with T, G with C, the spiral turning, slowly, gently, in the quiet dark of the nucleus, 320 million turns, all of them patient, all of them still. The atoms of your DNA, once inside stars, now inside you, here, at rest, in the night. There is nothing to do, nothing to remember, nothing to hold. The molecule is doing its work. The cell is doing its work. The body is doing its work. And you can simply drift. A with T, G with C, the helix turning, slowly, in the quiet dark. You are safe. You are warm. The ancient molecule is doing its ancient work. The body is at rest. The night is deep and still. And you are drifting, gently, like the long strand of DNA, wound quietly around itself, patient, still, at rest. Good night.